Dear learners, in this video we are going to study chapter 10, that is cell cycle and cell division. So read with us because reading is the first step towards a good learning. Here we start. Are you aware that all organisms, even the largest, start their life form as a single cell? You may wonder how a single cell then goes on to form such large organisms. Growth and reproduction are characteristics of cells, indeed of all living organisms. All cells reproduce by dividing into two, with each parental cell giving rise to two daughter cells. Each time they divide, these new formed daughter cells that can themselves grow and divide, giving rise to a new cell population that is formed by the growth and division of a single parental cell and its progeny. In other words, such cycles of growth and division allow a single cell to form a structure consisting of millions of cells. Cell cycle. Cell division is a very important process in all living organisms. During the division of a cell, DNA replication and cell growth also takes place. All these processes, it means cell division, DNA replication and cell growth, hence have to take place in coordinated way to ensure correct division and formation of progeny cells containing intact genomes. The sequence of events by which a cell duplicates its genomes and sizes the other constituents of the cell and eventually divides into two daughter cells is termed as cell cycle. Although cell growth in terms of cytoplasmic increase is a continuous process, DNA synthesis occurs only during the one specific stage in the cell cycle. The replicated chromosomes DNA are then distributed to daughter nuclei by a complex series of events during cell division. These events are themselves under genetic control. Next is phases of cell cycle. A typical eukaryotic cell cycle is illustrated by human cells in culture. These cells divide one in approximately every 24 hours. However, this duration of cell cycle can vary from organism to organism and also from cell type to cell type. Yeast, for example, can progress through the cell in only about 90 minutes. The cell cycle is divided into two basic phases: interphase, M phase. It means mitosis phase. The M phase represents the phase when the actual cell division or mitosis occurs, and the interphase represents the phase between two successive mitosis phases. It is significant to note that in 24-hour average duration of a cell cycle of a human cell, cell division proper lasts for only about an hour. The interphase lasts for more than 95% of the duration of cell cycle. The mitosis phase starts with the nuclear division, corresponding to the separation of daughter chromosomes, karyokinesis, and usually ends with division of cytoplasm, cytokinesis. The interphase, so called the resting phase, is called a time during which the cell is preparing for the division by undergoing both cell growth and DNA replication in an orderly manner. The interphase is divided into three further phases: Z1 phase, gap 1, S phase, synthesis, and G2 phase, that is gap 2. G1 phase corresponds to the interval between the mitosis and initiation of DNA replication. During G1 phase, the cell is metabolically active and continuously grows but does not replicate its DNA. S or synthesis phase marks the period during which DNA synthesis or replication takes place. During this time, the amount of DNA per cell doubles. If the initial amount of DNA is denoted as 2C, then it increases to 4C. However, there is no increase in the chromosome number. If the cell had diploid for 2n number of chromosomes at G1, even after S phase, the number of chromosomes remain the same. It means 2n. In animal cells, during S phase, DNA replication begins in the nucleus and centriole duplicates in the cytoplasm. During the G2 phase, proteins are synthesized in preparation for mitosis while cell growth continues. Some cells in adult animals do not appear to exhibit division, example heart cells, and many other cells divide only occasionally as needed to replace cells that have been lost because of injury or cell death. These cells that do not divide further exit G1 phase to enter an inactive stage called quiescent stage, G0, of the cell cycle. Cells in this stage remain metabolically active but no longer proliferate unless called on to do so, depending on the requirement of an organism. In animals, mitotic cell division is only seen in diploid somatic cells. However, there are few exceptions to this where haploid cells divide by mitosis. For example, male honeybees. Against this, the plants can show mitotic divisions in both haploid and diploid cells. From your recollection of examples of alternation of generations in plants, chapter 3, identify plant species and stages which mitosis is seen in haploid cells. Mitosis phase. This is most dramatic period of cell cycle involving a major reorganization of virtually all components of the cell. Since the number of chromosomes in the parent and progeny cells is the same, it is also called as equational division. For convenience, mitosis has been divided into 4 stages of nuclear division. Karyokinesis. It is a very essential to understand the cell division is progressive process and very clear-cut lines cannot be drawn between various stages. Karyokinesis involves following 4 stages: prophase, metaphase, anaphase, and telophase. Prophase. Prophase, which is the first stage of karyokinesis of mitosis, follows the S and G2 phases of interphase, and S and G2 phases of the new DNA molecule formed are not distinct but intertwined. Prophase is marked by the initiation of condensation of chromosomal material. The chromosomal material becomes untangled during the process of chromatin condensation. The centrosome, which had undergone duplication during S phase of interphase, now begins to move towards opposite poles of the cell. The completion of prophase can thus be marked by following characteristic events. First, chromosomal material condenses to form compact mitotic chromosomes. Chromosomes are seen to be composed of 2 chromatids attached together at the centromere. Second, a centrosome which had undergone duplication during interphase begins to move towards opposite poles of the cell. Each centrosome radiates out microtubules called esters. The 2 esters together with spindle fibers form mitotic apparatus. Cells at the end of prophase, when viewed under the microscope, do not show Golgi complexes, endoplasmic reticulum, nucleolus, and nuclear envelope. Metaphase. The complete disintegration of the nuclear envelope marks the start of second phase of mitosis. Hence, the chromosomes are spread through the cytoplasm of the cell. By this stage, condensation of chromosomes is completed and they can be observed clearly under the microscope. This is the stage at which morphology of chromosomes is most easily studied. At this stage, metaphase chromosome is made up of 2 sister chromatids which are held together by the centromere. Small disc-shaped structures at the surface of the centromeres are called kinetochores. These structures serve as the site of attachment of spindle fibers to the chromosomes that are moved into position at the center of the cell. Hence, the metaphase is characterized by all chromosomes coming to lie at the equator with 1 chromatid for each chromosome connected by its kinetochore to spindle fibers from one pole and its sister chromatid connected by its kinetochore to spindle fiber from the opposite pole. The plane of alignment of the chromosomes is metaphase is referred to as metaphase plate. The key features of metaphase are: first, a spindle fiber attached to kinetochores of the chromosome. Second, chromosomes are moved into the spindle equator and get aligned along the metaphase plate through spindle fibers to both poles. Next is anaphase. At one set of anaphase, each chromosome arranged at the metaphase plate is split simultaneously, and the 2 daughter chromatids, now referred to as daughter chromosomes of the future daughter nuclei, begin their migration towards the 2 opposite poles. As each chromosome moves away from the equatorial plate, the centromere of each chromosome remains directed towards the pole and hence is at the leading edge, with the arms of chromosomes trailing behind. Thus, anaphase stage is characterized by the following key events: centromeres split and chromatids separate. Chromatid move to opposite poles. Telophase. At the beginning of final stage of karyokinesis, it means telophase. The chromosomes that have reached their respective poles decondense and lose their individuality. The individual chromosomes can no longer be seen, and each stage of chromatin material tends to collect at each of the 2 poles. This is a stage which shows the following key events. First is chromosome cluster at opposite spindle poles and their identity is lost as discrete elements. Second is a nuclear envelope develops around the chromosome clusters as each pole forming 2 daughter nuclei. Say third, nucleolus, Golgi complex and ER reform. Cytokinesis. Mitosis accomplishes not only the segregation of duplicated chromosomes into daughter nuclei, karyokinesis, but the cell itself is divided into two daughter cells by the separation of cytoplasm called cytokinesis, at the end of which cell division gets completed. In an animal cell, this is achieved by appearance of furrow between the plasma membrane. The furrow gradually depends on the ultimately joins the center, dividing the cell cytoplasm into two. The plant cell, however, are enclosed by a relatively inextensible cell wall. Therefore, they undergo cytokinesis by a different mechanism. In plant, cell wall formation starts in the center of cell and grows outward to meet the existing lateral walls. The formation of new cell wall begins with the formation of a simple precursor called cell plate that represents the middle lamella between the walls of two adjacent cells. At the time of cytoplasmic division, organelles like mitochondria and blasts get distributed between the two daughter cells. In some organisms, karyokinesis is not followed by cytokinesis, as a result of which multinucleate condition arises, leading the formation of syncytium liquid endosperm in coconut. Significance of mitosis. Mitosis or the equational division is usually restricted in diploid cells only. However, in some lower plants and some social insects, haploid cells also divide by mitosis. It is very essential to understand the significance of this division in the life of an organism. Are you aware of the sample where you have studied about haploid and diploid insects? Mitosis usually results in the production of diploid daughter cells with identical genetic complement. The growth of multicellular organisms leads to mitosis. Cell growth results in disturbing the ratio between the nucleus and the cytoplasm. It therefore becomes essential of the cell divide to restore the nuclear cytoplasmic ratio. A very significant contribution of mitosis is cell repair. The cells of the upper layer of the epidermis, cells of the lining of the gut, and blood cells are being constantly replaced. Mitotic divisions in the meristematic tissues, the apical and the lateral cambium, result in the continuous growth of plants throughout their life. Meiosis. The reproduction of offspring by the sexual reproduction includes fusion of two gametes, each with a complete haploid set of chromosome. Gametes are formed from the specialized diploid cell. This is a specialized kind of cell division that reduces the chromosome number by half, results in the production of haploid daughter cells. This kind of division is called meiosis. Meiosis ensures the production of haploid phase in the life cycle of sexually producing organism, whereas fertilization restores the diploid phase. We come across meiosis during gametogenesis in plants and animals. This leads to the formation of haploid gametes. The key features of meiosis are as follows. First, meiosis involves two sequential cycles of nuclear and cell division called meiosis first and meiosis second, but only a single cycle of DNA replication, meiosis first, is initiated after the parental chromosome have replicated to produce identical sister chromatids at the S phase. Third is meiosis involves pairing of homologous chromosomes and recombination between non-sister chromatids of homologous chromosomes. Four haploid cells are formed at the end of meiosis second. Meiotic division can be grouped under the following phases: meiosis first, prophase first, meiosis second, prophase second, meiosis first, metaphase second, anaphase first, anaphase second, telophase first, telophase second. Meiosis first, prophase first. Prophase of the first meiotic division is typically longer and more complex when compared to prophase of mitosis. It has been further subdivided into the following 5 phases based on the chromosomal behavior. It means leptotene, zygotene, pachytene, diplotene, and diakinesis. During leptotene stage of the chromosomes between gradually visible under the light microscope, the compaction of chromosomes continues throughout leptotene. This is followed by second stage of prophase, first called zygotene. During this stage, chromosomes start together, and this process of association is called synapses. Such chromosomes are called homologous chromosomes. Electron micrographs of this stage indicate that chromosome synapses is accompanied by the formation of complex structure called synaptonemal complex. The complex formed by a pair of synapse homologous chromosomes is called bivalent or a tetrad. However, these are more clearly visible at the next two stages. The first two stages of the prophase first are relatively short-lived compared to the next stages, that is pachytene. During this stage, the four chromatids of each bivalent chromosomes become distinct and clearly appear as tetrads. These stages characterized by the appearance of recombination nodules, the sites at which the crossing over occurs between the non-sister chromatids of the homologous chromosomes. Crossing over, the exchange of genetic material between two homologous chromosomes. Crossing over is an also enzyme mediated process, and an enzyme involved is called recombinase. Crossing over leads to combination of genetic material on the two chromosomes. Recombination between homologous chromosomes is completed by the end of Pech18, leaving the chromosomes linked at the sites of crossing over. The beginning of the diplotin is recognized by the dissolution of the synaptonemal complex and the tendency of the recombined homologous chromosomes of the bivalents to separate from each other, except at the sites of crossovers. The X-shaped structures are called chiasmata. In oocytes of some vertebrate, diplotin can last for months or years. The final stage of meiotic prophase is diakinesis. This is marked by the terminalization of chiasmata. During the phase, the chromosomes are fully condensed and the meiotic spindle is assembled to prepare the homologous chromosomes for separation. By the end of diakinesis, the nucleolus disappears and the nuclear envelope also breaks down. Diakinesis represents the transition to metaphase. Metaphase first. The bivalent chromosomes align on the equatorial plate. The microtubules from the opposite poles of the spindle attach to the kinetochore of homologous chromosomes. Anaphase first. The homologous chromosomes separate while sister chromatids remain associated at their centromeres. Telophase first. The nuclear membrane and nucleolus reappear. Cytokinesis follows, and this is called as dyad of cells. Although in many cases the chromosomes do undergo some dispersion, they do not reach the extremely standard stage of interphase nucleus. The stage between the two meiotic division is called interkinesis and generally short-lived. There is no replication of DNA during interkinesis. Interkinesis is followed by prophase second, a much simpler prophase than prophase first. Meiosis second. Prophase second. Meiosis second is initiated immediately by cytokinesis, usually before the chromosomes are fully elongated. In contrast to meiosis second resembles a normal mitosis. The nuclear membrane disappears by the end of prophase second. The chromosomes again become compact. Metaphase second. At this stage, the chromosomes align at the equator and the microtubules from opposite poles of the spindle get attached to the kinetochores of sister chromatids. Anaphase second. It begins with the simultaneous splitting of centromere of each chromosome, which was holding the sister chromatids together, allowing them to move toward opposite poles of the cell by shortening the microtubules attached to kinetochores. Telophase second. Meiosis ends with telophase second, in which the two groups of chromosomes once again get enclosed by a nuclear envelope. Cytokinesis follows, resulting in the formation of tetrad of cells, 4 haploid daughter cells. Next is significance of meiosis. Meiosis is the mechanism by which conservation of specific chromosome number of each species is achieved across the generations in sexually reproducing organisms. Even though the process paradoxically results in the reduction of chromosome number by half, it also increases the genetic variability in the population of organism from one generation to the next. Variation are all very important for the process of evolution. Next, come to the summary. According to the cell theory, cell arise from pre-existing cells. The process by which this occurs is called cell division. Any sexually reproducing organism starts its life cycle from a single cell zygote. Cell division does not stop with the formation of mature organism but continues throughout its life cycle. The stages through which a cell phase from one division to the next is called the cell cycle. The cell cycle is divided into two phases called interphase and period of preparation of cell division, and second mitosis, and phase, the actual period of cell division. Interphase is further divided into G1 and G2. G1 phase is period when the cell grows and carries out normal metabolism. Most of the organelle duplication also occurs during this phase. As phase marks the phase of DNA replication and chromosome duplication. G2 phase is the period of cytoplasmic growth. Mitosis is also divided into 4 stages, namely prophase, metaphase, anaphase, and telophase. Chromosomes and condensation occurs during prophase. Simultaneously, the centrioles move to the opposite poles. The nuclear envelope and nucleolus disappears, and the spindle fibers start appearing. Metaphase is marked by the alignment of chromosomes at the equatorial plate. During the anaphase, at the centromeres divide and the chromatids start moving towards the two opposite poles. Once the chromatids reach the two poles, the chromal Chromosomal elongation starts nucleolus and the nuclear membrane repair. This stage is called telophase. Nuclear division is then followed by the cytoplasmic division and is called cytokinesis. Mitosis, this is equational division in which the chromosome number of the parent is conserved in the daughter cell. In contrast to mitosis, meiosis occurs in diploid cells which are destined to form gametes. It is called the reduction division since it reduces the chromosome number by half while making the gametes. In sexual reproduction, when the two gametes fuse, the chromosome number is restored to the value in the parent. Meiosis is divided into two phases, meiosis first and meiosis second. In the first meiotic division, the homologous chromosomes pair to form bivalents and undergo crossing over. Meiosis first has a long prophase which is divided further into 5 phases. These are leptotene, zygotene, pectine, diplotin, and diakinesis. During metaphase, first the bivalents arrange on the equatorial plate. This is followed by interphase first, in which homologous chromosomes move to the opposite poles with both their chromatids. Each pole receives half the chromosome number of the parent cell. In telophase second, the nuclear membrane and the nucleolus reappear. Meiosis second is similar to mitosis. During the anaphase second, the sister chromatids separate. Thus, at the end of meiosis, four haploid cells are formed. Here, the chapter is finished. So, bye.